Good morning, dear students. Today we will study gametogenesis. So, in order to maintain and propagate species in human beings, there is a process of fertilization in which the mature male gamete called spermatozoa fuses with the female gamete called mature ovum so that a single cell stage called zygote will develop embryo into a fetus. So, gametogenesis is the process of development and maturation of male and female gametes. In case of females, it is known as oogenesis, and in case of males, it is known as spermatogenesis. So far, we know that the male gametes are known as spermatozoa and they are present in testes, and in case of females the mature female gametes are known as mature ova and they are present in the cortical region of the ovaries. Now let us see from where do these gametes develop. Okay, so these gametes develop from the primordial germ cells. At second week of intrauterine life, the primordial germ cells develop from epiblast. We know that the trilaminar germ disc has epiblast, intraembryonic mesoderm, and the hypoblast. So the pluripotent cells of the pluripotent stem cells will give rise to the epiblast close to the extraembryonic mesoderm, and from these epiblast cells develops the primordial germ cells. Okay, so the gametes develop from primordial germ cells, and these primordial germ cells develop from epiblast at second week of intrauterine life. Okay, now at third week, as you can see in this diagram, at third week, these primordial germ cells will migrate to the dorsal wall of the yolk sac close to the allantoic canal. Okay, so the primordial germ cells have reached dorsal wall of yolk sac at third week of intrauterine life. After that, these germ cells will migrate to the dorsal mesentery of developing gut. Okay, so at fifth week, they will migrate from the dorsal mesentery of the developing gut into the developing gonads. Okay, so by the end of fifth week, the primordial germ cells will appear in the developing gonads. In case of males, they will appear in testes. In case of females, they will reach the cortical region of the ovaries. Now, when the germ cells reach the developing gonads, initially they will undergo rapid mitotic division so that there is increase in the number of germ cells. These germ cells have diploid number of chromosomes, means 44 autosomes, and 2 sex chromosomes are present initially. As they undergo mitotic division, there is rapid increase in the number of the germ cells. It is followed by meiotic division. The first meiotic division is reduction division, so it causes reduction in the number of chromosome, so that now the cells will become or they will have haploid number of chromosomes. Okay, so what we have seen till now, there is development of primordial germ cells from epiblast. They will first migrate into the dorsal wall of the yolk sac at third week, then they will reach the dorsal mesentery. From the dorsal mesentery, they will migrate into the developing gonads by fifth week. After reaching the gonads, they will undergo mitotic division to increase in the number, and then it will undergo meiotic division so that there is reduction in the number of chromosomes. Thus, we can say that the process of maturation of primordial germ cell into mature gametes is known as gametogenesis. In case of males, it is known as spermatogenesis, and in case of females, it is known as oogenesis. The process of maturation of primordial germ cell from primary oocyte to mature ova in females is known as oogenesis. In the oogenesis, first meiotic division occurs, which is heterotypical reduction division, and it is followed by second meiotic division. It is homotypical and it is more or less similar to mitosis. So we have seen that the primordial germ cells have entered into the cortical region of the ovary, undergoes mitotic division, it forms oogonia. So at third month of intrauterine life, these oogonia will start enlarging and they will differentiate into primary oocyte. By seventh month, all the oogonia are transformed to primary oocyte, and these primary oocytes will enter into prophase of first meiotic division. So here two processes are taking place simultaneously. Meiotic division is occurring and simultaneously there is maturation of the follicle in the cortical region of the ovary. So we will first study the maturation of the follicle and then we'll see how the meiotic division is taking place. So uh, here we know that there is primary oocyte. Okay, so this is the primary oocyte. It has eccentric nucleus and it is surrounded by flat epithelial follicular cells, which is in turn surrounded by the stromal cells of the ovary. So at this stage, the follicle is known as primordial follicle. So in the cortex of the ovary, during during the ovarian cycle, which occurs after puberty, after the puberty, during each menstrual cycle, the maturation of the follicles occur. Okay, what we have seen till now, third month oogonia will start converting to primary oocyte. At seventh month, all the oogonia are transferred to primary oocyte. This primary oocyte will enter into the prophase of first meiotic division at seventh month. Now at birth the prophase of first meiotic division will get arrested into prolonged resting phase known as diktiotin phase. And why there is arrested prophase of first meiotic division after birth? Because oocyte maturation inhibiting hormone is secreted by the follicular cells. So at the time of birth, we have about 80,000 such primordial follicles. Okay, at the time of birth, 80,000 primordial follicles are there. Now, at the time of puberty, only 40,000 primordial follicles or the follicles are persisting and rest of the follicles will degenerate. And after puberty, menstrual cycle begins. So during each menstrual cycle, about 5 to 12 such primordial follicles in the cortex of the ovary will start maturing, but only one follicle will mature completely. It is known as Graafian follicle, and at the time of ovulation, it will shed off oocyte. Okay, so the primordial follicles are formed and at the time of birth there are 80,000 follicles. At the time of puberty we have about 40,000 follicles, and after puberty during each menstrual cycle about 5 to 12 such primordial follicles will start developing or they will start maturing, but only one follicle will mature completely and it will form a mature Graafian follicle, which during the process of ovulation will shed off oocyte. Now we will see the process of maturation. So how the maturation of the follicle occurs. So the primary oocyte will enlarge in size. Initially it was 35 micron. Now it enlarges to about 140 micron. Initially the follicular cells surrounding the primary oocyte were flat epithelial cells. Then they will become cuboidal, and now the primordial follicle is known as primary follicle, in which there is enlarged primary oocyte surrounded by the follicular cells which are cuboidal. Okay, Later on, the follicular cell, initially there was single layer, but now it will become multi-layered follicular cell. Okay, so there is multiple layer of follicular cells surrounding the primary oocyte, and we can see in between the primary oocyte and the multi-layered follicular cell we have another membrane, amorphous glycoprotein layer is there, and it is known as zona pellucida. So this zona pellucida has many functions. During fertilization, it prevents polyspermia. During implantation, it prevents ectopic pregnancy. But if we restrict to oogenesis, the function of the zona pellucida is to provide nutrition by diffusion. The follicular cells will provide nutrition to the primary oocyte by diffusing through the zona pellucida. So this zona pellucida is developing from both primary oocyte as well as from the follicular cells. At the site of primary oocyte, multiple microvilli are there which will give it striated appearance. Okay, so this zona pellucida has striated appearance. So there is primary oocyte surrounded by zona pellucida, which is surrounded by multi-layered follicular cell, which is surrounded by the stromal cells of the ovary. After this, small fluid-filled cavities will appear in the multi-layered follicular cells. These fluid-filled cavities will coalesce to form a large cavity known as antrum folliculi, and the fluid present in this antrum folliculi is known as Leaker folliculi. Okay, so from the secondary follicle in which the cavities are there, the follicle is now converted to mature Graafian follicle. Now let us see the structure of mature Graafian follicle. So in the mature Graafian follicle, we have secondary oocyte. The cell membrane of secondary oocyte is known as vitelline membrane. Then the space between the zona pellucida, so this is the zona pellucida and this is the vitelline membrane. So the space in between these two is known as perivitelline space, which contains the first polar body. Then the follicular cells will arrange themselves in two layers. The layer of follicular cells surrounding the secondary oocyte is known as cumulus ovaricus. So this is cumulus ovaricus, which will later give rise to corona radiata. And the second layer of the follicular cell, which is present at the periphery, is known as membrana granulosa. So this layer of follicular cell is known as membrana granulosa. This is then surrounded by stroma of the ovary, which will again be converted into two layers. So the inner layer of the stroma of the ovary is known as theca interna. So this theca interna is a cellular layer, it is vascular layer, and the outer layer is known as tunica externa, or it is also known as theca externa. So it is the fibrous stromal layer. So this is all about the development of follicles, maturation of the follicle. Initially primordial follicle is there, it is converted to primary follicle, then it converts into the secondary follicle, and the secondary follicle then develops into a mature Graafian follicle, and we have seen all the changes occurring. Now simultaneously meiotic division is also occurring. So we have seen that the primary oocyte will start the first meiotic division in prenatal life. Okay, at 7th month, the primary oocyte will enter into the prophase of first meiotic division. After birth, there is release of oocyte maturation inhibiting hormone by the follicular cells. So it will arrest the prophase of first meiotic division in diktiotin stage. Diktiotin stage is a prolonged resting phase. Okay, so the first meiotic division at birth is arrested in diktiotin phase of prophase of first meiotic division. Now after puberty, when the menstrual cycle is beginning, when the follicles have started maturing, at this time, at the time of puberty, just before ovulation, the primary oocyte will, and it will complete the first meiotic division. Okay, the primary oocyte will complete the first meiotic division just before ovulation, and as it is first meiotic division, it is reduction division. We know that the primary oocyte has diploid number of chromosome, so when it will divide mitotically, it will give rise to two daughter cells, secondary oocyte and first polar body. So this is the secondary oocyte and the first polar body. As it is first meiotic division, it is reduction division, and there is equal distribution of chromosomal material. So both the daughter cells will have haploid number of chromosome. Initially, in the primary oocyte, 44 autosome and a pair of X chromosome or the sex chromosomes were present. Now after first meiotic division, the secondary oocyte and the first polar body both are having haploid number of chromosome, means 22 autosome and 1 X chromosome. But here we can see that the cytoplasm is unequally distributed. So the larger cell, which is known as secondary oocyte, gets abundant maternal cytoplasm. So it is larger cell. And the smaller cell, which is known as first polar body, though it has equal amount of chromosomal material, though it has haploid number of chromosome, but it has scanty cytoplasm. So it is smaller in size and it is present in perivital end stage. So during the process of oogenesis, we have seen that there is meiosis taking place and simultaneously there is maturation of the follicle also. So how the follicle will mature? There is initially primordial follicle which will get converted to primary follicle. Primary follicle, after the advent of the spaces, is converted to secondary follicle. Secondary follicle is then converted to a mature Graafian follicle. And how the meiosis occurs? The meiosis, first meiotic division, will start at 7th month of intrauterine life. The primary oocyte will enter into the prophase of first meiotic division, but at birth it is arrested in dictyotin stage of prophase of first meiotic division because oocyte maturation inhibiting hormone is secreted by the follicular cells. Now just before ovulation, the first meiotic division is completed and secondary oocyte and first polar body is formed. Now, after ovulation, within 24 hours after ovulation, if fertilization occurs, then the secondary oocyte will complete its second meiotic division. Okay, means what happens during ovulation? The secondary oocyte is shed off by the rupture of Graafian follicle, and this secondary oocyte is arrested in the metaphase of second meiotic division. It is arrested in metaphase of second meiotic division, and only fertilization occurs, then the second meiotic division is completed and the secondary oocyte is now dividing mitotically to form two daughter cells. The larger daughter cell, which is having haploid number of chromosome and abundant cytoplasm, is known as mature ovum, and the second smaller daughter cell, which is having haploid number of chromosome but scanty cytoplasm, is known as the second polar body. We'll study ovarian cycle in detail in our next session, but now we will just see what is ovulation. So here you can see initially there is primordial follicle, it will develop into primary ovarian follicle. Then with the advent of lacunase, it is converted to secondary follicle. Secondary follicle will then develop into a fully mature follicle which is known as Graafian follicle. So this is the Graafian follicle. At the time of ovulation. Now what is ovulation? Ovulation is the process where a fully formed Graafian follicle ruptures. A fully formed mature Graafian follicle ruptures from the surface of ovary to release the secondary oocyte. This process is known as ovulation. Now, when the secondary oocyte is released during ovulation, it is arrested in the metaphase of second meiotic division. So this secondary oocyte is arrested in the metaphase of second meiotic division. It will reach the ampulla of the fallopian tube, and if fertilization occurs within 24 hours after ovulation, then the second meiotic division is completed, and this secondary oocyte will give rise to two daughter cells. The larger daughter cell, which has haploid number of chromosome and abundant maternal cytoplasm, is known as mature ova, and the second polar body, which has scanty cytoplasm and haploid number of chromosome, is released in the perivitelline space. So when the ova is released during ovulation, we can see there is secondary oocyte, there is first polar body, and it is surrounded by cumulus ovaricus. So this cumulus ovaricus will later give rise to the corona radiata. Okay, so this is when fertilization occurs, there is completion of second meiotic division. But if fertilization does not occur, then the secondary oocyte will degenerate. Now what will happen to the Graafian follicle? The Graafian follicle, now if fertilization occurs, if implantation occurs, if there is pregnancy, then it is converted to corpus luteum. It has radiating blood vessels, but if pregnancy does not occur, then it will degenerate, it will form the corpus albicans. So this is the normal ovarian cycle in which the maturation of follicle occurs, and the Graafian follicle is converted to corpus luteum. Okay, now ovulation occurs 14 days prior to onset of next menstrual bleeding. How you will calculate the day of ovulation? It is occurring 14 days prior to the onset of next menstrual bleeding. So the menstrual cycle is calculated from first day of menstrual bleeding to the first day of next bleeding, and during pregnancy menstruation ceases. Now let us see the structure of secondary oocyte. So after ovulation, the Graafian follicle ruptures and it will release the secondary oocyte. This secondary oocyte is in arrested metaphase of second meiotic division. Now let us see its structure. It has eccentric nucleus. The nucleus is vesicular, and there is a centrosome which has a pair of centriole. Microlecithal deutoplasm is there. Then the cell membrane of the secondary oocyte is known as vitelline membrane. So this is the vitelline membrane. Then we have an amorphous glycoprotein membrane between the cumulus ovaricus and the secondary oocyte, and this is known as zona pellucida. So zona pellucida develops from both the oocyte as well as follicular cell, and it shows striations as microvilli are present from the side of oocyte. So this will help in diffusion of nutrition from the follicular cells to the developing oocyte. Now after zona pellucida, we have seen in the maturing follicle, we have a layer of follicular cells surrounding the oocyte known as cumulus ovaricus, and another layer, peripheral layer, is known as membrana granulosa. Now the layer of follicular cell which is surrounding the oocyte, it will give rise to corona radiata. Okay, so there are three layers. Outermost layer of follicular cells are forming corona radiata. Then the next layer is of zona pellucida. after zona pellucida, we have the perivitelline space, and the third layer is formed by the cell membrane of secondary oocyte, which is known as the vitelline membrane. And in the perivitelline space, we can see the first polar body. Now, if fertilization occurs after 24 hours of ovulation, then the secondary oocyte, which was initially arrested in metaphase of second meiotic division, will complete its second meiotic division, and it will give rise to two daughter cells. So the larger daughter cell, which has abundant maternal cytoplasm, is called mature ova, and the uh, smaller daughter cell, which has scanty cytoplasm and haploid number of chromosome, is known as second polar body. The second polar body is again released in the perivitelline space, and after that the mature ova will fuse with the spermatozoa during the process of fertilization, and it will give rise to zygote. Now we will study the process of spermatogenesis, that is maturation of the male gametes. So the primordial germ cells, which were derived from the epiblast or the pluripotent stem cells, will migrate through dorsal mesentery into the developing testis. And we can see the structure of testis here. The parenchyma of the testis is divided by septa into lobule, and in each lobule we get highly coiled structure called seminiferous tubule. Now if we take the section of seminiferous tubule and see under microscope, then we can see two types of cells within the seminiferous tubule. The first type of cell is called Sertoli cell, and the other variety is the germ cells in various stages. Now the Sertoli cell is a tall columnar cell extending from the basement membrane up to the lumen of the seminiferous tubule, and these Sertoli cells provide nourishment to the developing germ cells. They will provide support to them. They will engulf or phagocytose the residual body, and most important function of Sertoli cell is that it forms a blood testis barrier. We know that the spermatogenesis begins or it starts at puberty. So the developing germ cells may act as antigen. These germ cells may act as a antigen to the immune system of our body, and the antibodies may attack and anti-spermic action may occur. So in order to prevent all this, the Sertoli cells will form blood testis barrier, and these germ cells are kept in a secluded compartment until the process of spermatogenesis is completed. And once the spermatozoa is formed and it is matured, it is released into the lumen of seminiferous tubule. Okay, so this is all about the Sertoli cells. It provides nourishment, it provides support, then it phagocytose the residual bodies, and mainly it forms the blood testis barrier. It also secretes Mullerian inhibiting factor, which will inhibit the paramesonephros. Now coming to the second type of cell, it is the germ cells in various stages. So within the seminiferous tubule, the germ cells are arranged in various compartments, or they are arranged in various layers. So this is the outer layer in which we get the spermatogonia. These spermatogonia are derived from primordial germ cells by mitosis. So the spermatogonia are present in outer layer and they have diploid number of chromosomes. When the spermatogonia undergoes mitotic division, it forms the primary spermatocytes. So in the intermediate layer, again we have outer zone and inner zone. So in the outer zone of intermediate layer, we have the primary spermatocytes which have diploid number of chromosomes. Now these primary spermatocytes will undergo first meiotic division, which is a reduction division, and each primary spermatocyte will give rise to two secondary spermatocytes, and as it is first meiotic division, it is reduction division. So the secondary spermatocytes present in the inner zone of intermediate layer will have haploid number of chromosomes. Then in the inner layer, we have the spermatids. So the secondary spermatocyte will undergo second meiotic division and it will form the spermatids. Okay, so this is all about the seminiferous tubule and the structures or the cells present within the seminiferous tubules. Now we will study the process of spermatogenesis. So the spermatogenesis is the process of maturation of male gametes in the wall of seminiferous tubule, and the spermatogenesis begins at puberty, and there are three stages of spermatogenesis. The first stage is called spermatocytosis. In this spermatocytosis, mitotic division occurs so that primordial germ cells are forming many or numerous uh, germ cells. Okay, then the second step is meiosis. So first meiotic division is reduction division, and second meiotic division is similar to mitosis, and the last stage is spermiogenesis, in which there is conversion of spermatids into spermatozoa. So the entire process of spermatogenesis is divided in three stages. Now first we will begin with spermatocytosis. So the primordial germ cell will reach the testis, it will undergo mitotic division, and it will form dark type A spermatogonia. So the dark type A spermatogonia are the stem cells, and they will divide to form dark type A and light type A spermatogonia. The dark type A spermatogonia are reservoir, whereas the light type A spermatogonia will further divide mitotically and it will give rise to type B spermatogonia. So these type B spermatogonia will undergo mitotic division. About 4 generations of type B spermatogonia are formed by continuous mitosis, and then it is dividing to form the primary spermatocyte. So till this stage we have diploid number of chromosomes. So the primary spermatocyte has diploid number of chromosomes. So till this stage we can say it is the spermatocytosis, in which up till now mitotic division has occurred from primordial germ cell up to the primary spermatocyte, and these all cells have diploid number of chromosome, that is 44 autosome and XY sex chromosome. Now the first meiotic division will occur. So the primary spermatocyte will undergo first meiotic division, that is reduction division, and it will give rise to two secondary spermatocytes which are having haploid number of chromosome, means they have 22 autosome and they have either X or Y chromosome. Then the second meiotic division occurs. The secondary spermatocyte will divide into two spermatids. So these are two secondary spermatocytes. They will undergo second meiotic division and they will form two spermatids. So in all, four spermatids are formed from a single spermatogonia. Here we have to remember that may it be spermatogenesis or oogenesis, each daughter cell after first meiotic division, which is a reduction division, has haploid number of chromosome, but the DNA will replicate. So we have two strands of DNA and haploid number of chromosome in the daughter cells after first meiotic division, whereas after second meiotic division there are haploid chromosome and we have single strand of DNA. Then the third stage is spermiogenesis. Here there will be no cell division. There is only morphological transformation of spermatids into the spermatozoa. Now let us briefly and rapidly revise the process of spermatogenesis. So there are three stages: spermatocytosis, meiosis and spermiogenesis. In spermatocytosis, the primordial germ cells will form dark type A spermatogonia. type A will undergo mitotic division to form dark and light type A. The light type A will undergo mitotic division to form type B spermatogonia. Type B will again divide mitotically, and finally it will form the primary spermatocyte. So up till this stage we can say there are diploid number of chromosome as only mitotic division is occurring, and this process is called spermatocytosis. Then the next stage is meiosis. So after first meiotic division or reduction division from a single primary spermatocyte, two haploid secondary spermatocytes are formed. Now the second meiotic division occurs, in which the two secondary spermatocytes will give rise to four spermatids. So each secondary spermatocyte will give rise to two spermatids. So in all, from a single spermatogonia, four spermatids are formed. And the third process is spermiogenesis, in which there is no cell division, there is only morphological transformation of spermatids into spermatozoa. After the second meiotic division, spermatids are formed. These spermatids will not divide. There will be no further cell division, and only there will be morphological transformation of spermatid to spermatozoa. So these spermatids are having nucleus, they have Golgi apparatus, they have mitochondria, they have centrosome having a pair of centriole, and abundant cytoplasm is present in the spermatid. Now how this spermatid is converted to spermatozoa? So the Golgi apparatus contains acrosomal granules. So the vesicles of these acrosomal granules are present in the Golgi apparatus. So it will form a bilaminar head cap or acrosomal cap. Okay, so the Golgi apparatus will form the acrosomal cap or head cap along the anterior pole of the nucleus, and it will cover about two-thirds of the nucleus. Now posterior to the nucleus there is centrosome. The centrosome will divide into two centrioles, so they will limit the middle piece. So these centrioles will mainly form the middle piece, the axial filament. It will form the axial filament and the tail piece, and this posterior centriole will form the ring centriole. So it is limiting the middle piece. Okay, and the mitochondria, it will arrange itself spirally around the axial filament. Then the nucleus, it will evaginate the cytoplasm, but it is still covered by cell membrane, and it will form the head of the spermatozoa, and excess of cytoplasm is now extruded. So now let us see the structure of spermatozoa. So in the spermatozoa, this head is formed by the nucleus, and it contains haploid number of chromosomes. So when the nucleus evaginates cytoplasm and when it is still covered by the cell membrane, it is forming the head. The acrosomal cap or head cap covering anterior two-thirds of the head is formed by the Golgi apparatus, and it has the acrosomal granules within it. Then the neck contains the first centriole, which is present along the posterior pole of the nucleus or the head. It is present along the posterior pole of the head. Now the axial filament and the tail, it is formed by the centriole, and the posterior centriole will form a ring centriole. Then the mitochondria will arrange itself spirally to form the mitochondrial sheath. Okay, so this is the structure of spermatozoa, and the entire process of spermatogenesis from that is development from primordial germ cell up to the spermatozoa takes 64 days in man. So the process of spermatocytosis requires 16 days to complete. Meiosis 1 needs 8 days. Meiosis 2 takes 16 days to complete. Spermiogenesis occurs about 24 days after second meiotic division. The fertilizing capacity of the spermatozoa once it is ejaculated and once it enters into the female genital tract, it is about 24 to 48 hours. In single ejaculation, normally 200 to 300 million sperms are ejaculated or emitted, and if the sperm count is less than 20 million, then the male is called sterile. Coming to the clinical significance, so if there is complete lack of semen during ejaculation, it is called aspermia. Complete absence of sperm is called azoospermia. If the sperm count is low, if it's less than 20 million, it is called oligospermia. Then teratospermia is the production of morphologically abnormal sperms. And if the sperm motility is less than 50%, it is called asthenospermia. Again, if there is any deviation from the normal cell division, then it will lead to chromosomal anomalies. So there can be structural or numerical chromosomal anomalies if the cell division does not occur in normal way. We will quickly revise the difference between spermatogenesis and oogenesis. So the spermatogenesis occurs in male in the testis, and in oogenesis is occurring in female in ovaries. Then with each spermatogenesis, number of gametes are produced, and it is a lifelong process. Spermatogenesis starts at puberty and it occurs till death, whereas oogenesis has fixed amount of follicles. About only 400 to 500 mature follicles are present at the time of puberty, and after menopause there is no further production of gametes. Then the gametes per germ cell. Now a single spermatogonia will give rise to four spermatids or spermatozoa, and in oogenesis a single uh, oogonia will give rise only to a single mature ovum. Then the spermatogenesis begins at puberty, oogenesis begins in the prenatal life. Then what is the time of gamete formation? In case of males, it is a continuous process from the puberty till death, whereas oogenesis occurs once in a month. During each menstrual cycle, only one follicle is shed off. Then the fertility. Males remain fertile till death. The fertility in females stops after menopause. Then the gametes can be released anytime in males. In females, there is a monthly cycle, and only after the process of ovulation, the gamete is released. Then, if we see the meiotic division, it is uninterrupted. At puberty, the meiotic division will start in males and it will be completed. But in females, the meiotic division starts prenatally at 7th month. At birth, it is arrested in dictyotin of prophase 1 meiotic division. Then after puberty, just before ovulation, the first meiotic division is completed. Again, the secondary oocyte will remain arrested in metaphase of second meiotic division, and second meiotic division is completed only after fertilization. Then the germ-line epithelium is involved in production of gametes in males, whereas the germ-line will not produce gametes in the females. So these are some of the important differences in spermatogenesis and oogenesis.